Okay, so today we are starting chapter 13, the brain and cranial nerves. Starting with just an overview of brain anatomy, there are four major regions to the brain. There is the cerebrum, which are the top lobes. When you think of your brain, kind of you're thinking about your cerebrum usually. It has the lobes that we know. There's the diencephalon, which is deep to that. There's the brainstem, which is a little bit lower, and there's the cerebellum, which sits on the bottom on the back. Whenever we think about our brain, we always also think about the folds that the brain has. Those folds are called the gyri. The sulci are the depressions. So in between those folds are the ridges. Those are the sulci, just the normal shallow ones. And the deeper ones are called the fissures of the brain. When we think about directions, you have anterior, which is rostral. If you remember that rostral means towards the nose or towards the front of the face, whereas caudal means towards the tail or towards the back. We'll look in just a second at brainal development, and we'll see that the central nervous system develops cranial to caudal meaning towards the tail, which is eventually the spinal cord. Here is just a picture, just a lateral view. You can see the frontal lobe, parietal, temporal, and occipital. That makes up your cerebrum. You have your cerebellum down here, as well as the brainstem, and then deep into here is the diencephalon. And there you go. You can see it breaks it up a little bit. So this is the region of the diencephalon. On top of that, so this is the inferior view. So superior to all of that would be your cerebrum. That's your cerebellum. It's a little bit easier to see inferiorly. And then all of these yellow things are the cranial nerves. There are 12 of them. You will have to learn them. Okay, moving on. So here, now that we've taken away, we're looking at this mid-sagittal view. So if you remember, the mid-sagittal is taking a cross-section directly down the middle. So this is the mid-sagittal view of the brain. You can see the actual intricacies of the diencephalon. This is the corpus callosum that connects the two halves, the two hemispheres. This area right in here is your thalamus. Here is your hypothalamus, hypo meaning underneath. You have your pineal gland back here. You have your epithalamus back here as well. And then you get into your brainstem. You have your midbrain, your pons, and then the medulla connecting to the spinal cord. So looking at the development of the brain divisions, this process is called neurulation. It starts in the third week of embryonic development. The embryonic development is from the first through 8 weeks of development. And so after the egg has implanted into the uterus, that's when you start your embryonic development. So for this first 8 weeks, there's a lot of growth happening. And then in that third week, the beginning of the third week, neurulation starts. It's finished or almost finished by the end of that third week. What happens is that part of the embryo's ectoderm, that outer layer of development, is thickened into a neural plate, what's called a neural plate, and then beneath that is a structure called the notochord that induces that basically kind of triggers the neural plate to form the neural tube, which develops into the central nervous system. Sorry, it was hidden. So this notochord is a fetal structure, is an embryonic structure rather, that basically guides development. It allows for a central axis to be established that the development then happens around. That's why we all end up looking the same. We have 2 arms coming off of our shoulders, 2 legs off of our hips, and our head where it is, because we have in embryonic development this structure that regulates how things are going to be and kind of positions us. So I went to watch this YouTube video really quick about neurulation, just because I think it's easier.
The first event of neurulation is the formation of a thickened area of cells, called the neural plate. The neural plate forms at the cranial end of the embryo and grows in a cranial to caudal direction. The cranial or head end of the neural plate indicates the region of the future brain, and the narrower caudal or tail end represents the future region of the spinal cord. By the end of the third week of development, the lateral edges of the neural plate become elevated and move together to form the neural folds. The resulting space, created by the folding of the neural plate, is called the neural groove. The neural folds fuse together, and the neural plate transforms into the neural tube, the precursor to the central nervous system. Fusion of the neural tube usually begins in the middle of the embryo, extending in both cranial and caudal directions. During the closure of the neural tube, cells on the crest of the neural folds detach, forming a new cell population called the neural crest. These cells contribute to the formation of the peripheral nervous system. Once the neural tube has completely fused, the process of neurulation is complete.
Okay. So now let's look at that process just a little bit more detail. So the neural plate develops its neural groove. What's going to happen is those cells proliferate along the lateral plate margins, and they start to come up, and they form the neural folds, and their tips become the neural crests. As you can see here, your notochord again is that central axis dividing organization. You have your neural folds that start to develop. As they develop, they have a groove that comes between them. That's your neural groove. And then the tip there, the top there, is the crest. So you have the top of the hills and then the valleys. The neural groove deepens as the folds start to rise and start to come towards each other and start to connect. And the neural crest cells separate from the neural folds. As we saw, those become the PNS. By the end of the third week, the folds meet and form the neural tube surrounding the neural canal. As fusion starts to begin in the middle, then progresses superiorly and inferiorly. So cranially superiorly and caudally inferiorly. Openings at the cranial and caudal ends of the tube are sealed by the end of the fourth week. If they do not, then you have neural tube structures. So this is again how the central nervous system is going to be formed. This neural tube and the neural canal are the spinal cord and the spinal canal. There's the defects and anencephaly and spina bifida if those neuropores do not fully close. Your cranial and your caudal neuropores. So looking at brain development, further brain development, I'm just going to strengthen myself more. So from the late fourth week, from the cranial neural tube, you have the forebrain, the midbrain, and the hindbrain that are all having to develop. Those are all structures that are developing. Those are your prosencephalon, mesencephalon, and rhombencephalon. Prosencephalon is the forebrain. Pros means fore or first. Then you have your mesencephalon, mes meaning middle, and then the rhombencephalon. The rhomb refers to the rhomboid shape of this. And then attached, again, continuous with that is the spinal cord. From the fifth week, so you start with just these three structures, and then that starts to differentiate even more into even more different structures. So your telencephalon will eventually become the cerebrum, again that part that sits right on top. The diencephalon will differentiate into the thalamus, the hypothalamus, and the epithalamus, and we still refer to all of those structures as the diencephalon collectively. The mesencephalon, metencephalon, and myelencephalon will differentiate to become both your midbrain, all the structures, the midbrain, the pons, and the medulla, so your brainstem, and then your cerebellum as well. I prefer this chart in your book, or from your book. So you start with just this one neural tube that forms in the process of neurulation, and then as development continues, it starts to differentiate into more and more distinct structures. So the primary brain vesicles are just these three, the prosencephalon, mesencephalon, rhombencephalon, and then you have secondary ones, the telen, diencephalon, meden, and myelin. And then they start to become even more adult structures, the more their final shape. The telencephalon is the cerebrum. The diencephalon is made up of the three epi, hypo, and thalamus. The mesencephalon, metencephalon, and myelencephalon, like I said, are going to become different structures of the brainstem and the cerebellum. Okay, this was, by the way, table 13.1 in your book. So continuing with development. So after all of that is starting to finish, we get into our fetal development. That's beyond from 8 weeks until birth. you are considered a fetus. It is fetal development. So the brain continues to develop. The telencephalon starts to envelop the diencephalon and develop surface folds. So remember, the telencephalon is what's going to become the cerebrum, the part with the lobes that we think of, and all the folds and the gyri, the gyri and the sulci. So the diencephalon is deep to that, and the telencephalon is going to form over top of it. The folds allow the brain to fit within the cranial cavity. It's kind of like how our intestines have to fold to fit into our abdomen. The brain has to have all of those folds to contain its larger surface area. By birth, most gyri and sulci are present. It's actually very interesting. There's a correlation and a connection between brain size at birth and development. So when we look at other primates, chimpanzees, they tend to have a longer, or they tend to be more developed at birth, because they really, if you think just evolutionarily, they are required to be more developed. There are numerous examples of mammals being born and then within a few hours having to be walking, running, keeping up with herds. And part of that is because their brains are allowed to develop larger brains, that they can develop more in utero, so that way they can be more ready, more functionally ready to care for themselves when they come out. Because of our structure, our female structure of the pelvis, it's not large enough to accommodate an oversized brain. So that's why in humans, our infants tend to be very underdeveloped compared to other mammals and other primates, because we just don't stay in utero long enough to develop that stronger functional features, those stronger functional attributes. If we were allowed to stay in utero longer, our brains would develop more fully, but then they would be so large they wouldn't be able to pass through the birth canal. Sorry, that was a long explanation. Okay, so now getting into gray and white matter distribution. So that was, we kind of got through all the development of the brain. Now let's start to look at some more of the structural features of the brain. So the brain and the spinal cord are composed of both gray and white matter. Gray matter is made up of the neuron cell bodies, the dendrites, and the unmyelinated axons. White matter is going to be your myelinated axons. If you remember, myelin is always kind of a clear or a whitish kind of color because it has that high fat content, it has that high lipid content. So just remember that the lipid content kind of gives myelin its whitish appearance, so that's white matter, whereas gray matter is going to be the unmyelinated axons, the dendrites, and the cell bodies. Within the brain, we have both cortex and we have both superficial and deep features. So the superficial gray matter is called the cortex. It's the very kind of most outermost portion of your brain, whereas the nucleus are the deeper portions of gray matter. So both of them are gray matter. Just the cortex is going to be the superficial portion, and then the nucleus will be the deeper portions. And then in white matter, it's organized into bundles called tracts. So we'll see correlations between these names in the CNS versus the PNS as well. Okay, so here you can see this. So the gray matter is going to be superficially called the cortex, and deep it's considered the nuclei, the cerebral nuclei. And then in the white matter, you have your corpus callosum, which is connecting to the two sides. Remember, these are the tracts, these are the axons, the myelinated axons that are carrying information. So they're going to be your corpus callosum and then these internal capsule structures, whereas the gray matter is going to be all the cell bodies doing all the processing and integration. As you start to go into your cerebellum and you still have that same external gray matter, internal white matter, but then when we switch into the spinal cord, that switches. So in the spinal cord, you have outer white matter and inner gray matter. So in the cerebellum and the cerebrum, you have outer gray matter, inner white matter. In the spinal cord, you have outer white matter and inner gray matter. It's just flipped in the spinal cord. Okay, also in the brain are cranial meninges. You have probably heard of meningitis. You've probably been scared of that term before. So the meninges are three connective tissue layers covering the brain. They help to separate and support the soft tissue. They enclose and protect blood vessels supplying the brain. Remember, connective tissue, that's one of its features, is that it houses blood vessels. It helps contain and circulate cerebrospinal fluid that we'll look at. And from deep to superficial, there are three layers: the pia mater, the arachnoid mater, and the dura mater. Yeah, so these are just layers over the top of the brain. The pia mater is the innermost of the meninges. It adheres to the brain surface, so it is completely continuous with the surface of the brain. It is a thin layer of areolar connective tissue. The arachnoid mater lies external to the pia mater. So it is, if you think your pia mater is directly attached to your brain, this is slightly more superficial, is your arachnoid mater. It has what are called arachnoid trabeculae. You might remember that name from looking at bones. The spongy bone has the trabeculae. So the arachnoid trabeculae are extended through that subarachnoid space, the space between the arachnoid and the pia mater. That's the subarachnoid space. And there are the arachnoid trabeculae that are extended through there. Remember, the trabeculae are just, that's just a cross-hatching of structure, of protein structures. The subarachnoid space contains that cerebrospinal fluid. It is made of collagen and elastic fibers. So compared to the areolar connective tissue, collagen and elastic will give it a little bit more structure, a little bit more hardiness. It is deep to the dura mater, which is the most external one. And in between those is called the subdural space. So in between, you have your pia mater directly attached to the brain. Then you have the subarachnoid space. Then you have your arachnoid mater. And then in between the arachnoid mater and your dura mater is the subdural space. And that is a potential space that can fill with blood if a vein is ruptured. So what that means is that even though you have your arachnoid mater, your dura mater, and they're separate, there's actually, the space between them is only potential, meaning that it could separate, but only if something is going wrong. Ideally, there should really be no gap in there, except where it is structurally appropriate. Sorry, I just want to move myself around. Okay, so your dura mater is the tough outer membrane of your brain, covering your meninges. It is made of dense, irregular connective tissue. So the pia mater, the areolar connective tissue, a little bit softer, a little bit more fenestrated with holes. The arachnoid mater had the collagen and elastic fibers, whereas the dura mater has that dense, irregular connective tissue, and it's separated into two different layers. So it has both a meningeal layer and an outer periosteal layer. So the meningeal layer is the deeper layer of the dura, whereas the periosteal layer is the one that's fused to the cranial bones. They're usually fused, but in some areas they are separated to form the venal sinuses, the dural venous sinuses, that help to drain blood from, the brain. The epidural space is a potential space between the dura and the skull. So just like the subdural space is a potential space between the arachnoid and the dura mater, the epidural space is a potential space between the cranial bones and that outer periosteal layer of the dura mater. It does contain arteries and veins, so there are places where it's flowing. If one of those were to rupture, it would start to release blood into that space and put pressure on the brain. So here is just a view of those. This is the skin, and then you start to get into the periosteum, the bone, and then you start to have, from the bone to this periosteal layer of the dura mater, you have that epidural space. Then you have, again, just directly adhered to the periosteal layer is this meningeal layer. And then, hold on one second. And then you have the subdural space, the arachnoid mater, there's the arachnoid trabeculae, and then you have your pia mater as well, adhered directly to this cortex, the cerebral cortex. Okay, let's see. Yeah, and then there's the venous sinuses separating them, draining. Okay, the cranial dura septer. So there are sheets of dura mater that extend into the cranial cavity. They form partitions between the brain and help to provide support. Sorry, there's a cloud, so it's getting a little darker. These have several different names. The largest one is the falx cerebri, or sorry, I always say that, falx cerebri. It's the largest of the dural septa. It's located along the midline and then projects into the longitudinal fissures between the cerebral hemispheres. It has the superior sagittal sinus and inferior sagittal sinus. And then the tentorium cerebelli covers the cerebellum. So you have the falx cerebri and then your tentorium cerebelli. So cerebri referring to your cerebrum, the big one, and then cerebelli referring to your cerebellum. That will be confusing until you just start committing it to memory. The tentorium cerebelli separates the occipital and temporal lobes. So the occipital, the bottom one, and the temporal on the side. It separates those two lobes. It contains the transverse and the straight sinus and has a tentorial notch. So I think it's helpful, and when I am studying and reviewing for lectures, I usually have this app open that helps me to visualize. This is my anatomy app that I use. So when I'm going through the notes, I have the notes open. I also have the book open, and I have this app open. So when I'm looking at reading over structures, I can actually look at structures. So this is just the basics of the nervous system. Actually, if you want to, we can see. This is essentially all of your nerves. This is your full nervous system right here. Your spinal cord and brain highlighted as the central nervous system, and then all of those yellow ones are the peripheral nerves. I always like to point out, look how big you ever hear people have sciatica. Look how large that sciatic nerve is compared to all the other ones. There's no reason or no surprise that it gives you pain. Okay, but I only need this much. So looking at our brain, this outer layer right here, that's the dura mater, so I can hide that. Then you have your arachnoid mater. I'll hide that. It doesn't show the pia mater because it is directly adhered to the surface, so it's a continuous layer with the surface. But real quick, let's just look at the regions of the brain. So you can see all of the different folds, the gyri. You can see these little grooves, which are the sulci, and there are deeper fissures as well. Sorry, I always have to reset. That's the one thing about this app is that it's not really great for organizing or manipulating. So you have your different lobes. You have your frontal lobe, and you have your parietal lobe. You have your temporal lobe, and then back here you have your occipital lobe. There's your cerebellum, part of your cerebellum. This right in here, there's your pons. That's your medulla, or parts of your medulla, and then your brainstem. So just kind of giving you an idea about what your brain is organized as. So this right here, going back to what we were talking about, the cranial dura septa, this is your falx cerebri right here. You can see it goes all the way around, and if I can get control, if I select this and hide, you can see it is a full sheet of connective tissue that's separating them. And then also we have this tentorium cerebri or cerebelli, which is separating and dividing the. How do I want to do this? I'm going to do it this way. This tentorium cerebelli, which is separating and dividing the cerebellum from the cerebrum. Yeah. I don't think there's any others that I wanted to do. Oh yeah, the falx cerebelli then is this one that is dividing the cerebellum in half. And the diaphragm ocelli, I can't show you that one on this model. Okay, so we will probably come back to this because it is helpful, I think, to be able to see it that way. Okay, so here's just your book's view of that. Maybe this will make a little bit more sense now that you have seen that other model. So there are cavities within your brain as well, called the ventricles of the brain. They are lined with ependymal cells, and they contain cerebrospinal fluid. These help to connect with each other into the spinal cord central canal. So these are just, we'll see. There are 4 of them. There are 2 lateral ventricles, large cavities in the cerebrum separated by a medial partition called the septum pellucidum. There is the 3rd ventricle, so you have 2, and then they go right into 3rd, which is a narrow space in the middle of the diencephalon, connected to each lateral ventricle by an interventricular foramen. If you remember, a foramen is a hole, interventricular between the ventricles. And then you have your 4th ventricle, which is a sickle-shaped space between the pons and the cerebellum. So the cerebellum and part of the brainstem. It's connected to the 3rd ventricle by the cerebral aqueduct. An aqueduct is something that connects fluids, that drains fluids, connecting spaces. It opens directly to the subarachnoid space on the medial and lateral sides, and it narrows before merging with the central canal of the spinal cord. So all of these are essentially containing cerebrospinal fluid that is circulating through the brain and then back down into the spinal cord. Here is the book's visual representation of them. So you have these kind of large arching structures. These are your 2 lateral ventricles. You have your 3rd ventricle in the diencephalon, and then your 4th ventricle down here in the medulla, and the pons and the cerebellum. I wanted to show you all on here as well because I think that this is just a slightly better representation. Again, if I can work. To hide the layers, hide the brain, and then you can start to see, okay, so this right here, that is your lateral ventricle. So this whole thing is the lateral ventricle. And there are two of them. So if I hide this brain, so there's the second one. So there's one on each side. You can kind of see it right there. And then if we come in here, let's see, come in here and we hide our cerebellum, get rid of it completely, you can start to see the fourth ventricle. Okay, so you have your, let's go and get rid of this too. So you have your lateral ventricles on either side. You have your one, let me hide this, your one fourth ventricle right there. And then your third ventricle is up in your diencephalon. So we're going to zoom in. We're going to get rid of our thalamus. And there is our third ventricle. Get rid of this other side of the thalamus. There you go. And there's your third ventricle. And you can see it's connected through the cerebral aqueduct. And then your third ventricle, let's see if we can see its connections to the, yeah, that should be it. Yep, the interventricular foramen. So you have your lateral ventricles draining into your third ventricle, which connects down to your fourth ventricle, which leads to your spinal cord. Okay. What are those ventricles? What do they have? They have cerebrospinal fluid inside of them. So cerebrospinal CSF is a clear, colorless liquid surrounding the central nervous system. It circulates in the ventricles and the subarachnoid space, and it works to provide buoyancy. So it reduces the brain's weight by 95%. I thought something kind of shocking and scary in your book was that, you know, without the cerebrospinal fluid, your brain would just sink down through the foramen magnum, the hole in your skull. The weight would just make it collapse on itself. So the CSF really helps to make it a little bit more light. It offers protection as a cushion. It helps to, you know, reduce movement. If you think about how liquid suspension is a way that you can help to reduce things sloshing around. Even just a fetus developing in the uterus is surrounded by all that amniotic fluid to help reduce impact. It also provides environmental stability. So it transports nutrients and wastes and protects against fluctuations in ion concentration, sodium, potassium, calcium, chloride, glucose. It makes sure that all of those levels stay the same. Make sure any kind of pathogens don't get in. Or rather, CSF doesn't do that, but the blood-brain barrier does. We'll get into it. So the CSF is formed by the choroid plexus in each ventricle. The choroid plexus is a layer of ependymal cells, those ependymal cells, and the blood capillaries within the pia mater. So real quick again, just so you can see what it means. Like this. So there's our lateral ventricle. If we hide that, there is your choroid plexus. Okay, so just deep to the ventricle is that choroid plexus. Blood plasma is filtered through the capillaries in the pia mater and modified by those ependymal cells. So it has to pass through the blood capillary. It goes through the ependymal cells. It goes through translational transport so that it has to be modified within the ependymal cells, and then it gets released into the ventricles. So compared to plasma, cerebrospinal fluid is going to have more sodium and chloride and less potassium, calcium, and glucose than regular blood plasma would. This is just going to help make sure that proper neurological function can be maintained. In addition, the ependymal cells, secretions, and interstitial fluid from your subarachnoid space help make up to the CSF. So in addition to blood plasma being filtered, you also have secretions from the ependymal cells to help make up CSF. CSF is continuously being formed and reabsorbed. Excess CSF flows into arachnoid villi and drains into the dural venous sinuses, and that eventually returns into just circulating blood volume. An arachnoid granulation is a collection of these villi. So here you go. This is just looking at the choroid plexus, that structure I showed you on the application. So you have this section right here of a blood capillary. So this is your blood capillary with its ependymal cells surrounding it. And then in between is the pia mater, that thin pia mater. And so what's going to happen is that the blood pressure will force filtration through the cells, through the blood capillary layer, the pia mater, and then in between the ependymal, or through the ependymal cells into the central, the cavity of the ventricle. Okay, here is just your book's kind of representation of that. So production starting here, flowing out through the ventricles, the third ventricle, until it can be circulated around through that subarachnoid space. This is one of those arachnoid villi that allows fluid to be filtered out, or arachnoid villus, I guess. Okay, and then the blood-brain barrier. So functions of that blood-brain barrier helps to regulate substances entering the brain's interstitial fluid. It helps to prevent neuron exposure to harmful substances, drugs, wastes, abnormal solute concentrations. Even though some drugs can pass through there, lipids are freely able to pass. Alcohol can easily pass through there. And certain drugs, cocaine, can damage that blood-brain barrier. So that way, meaning that it won't be as effective. The blood-brain barrier is composed of specialized capillaries. They have endothelial cells that are connected by many tight junctions. So normally when we think of capillaries, we think they're there to enhance and to help for filtration, whereas these have a lot of those really tight junctions, so that and it has a thick basement membrane. The tight junctions, again, don't allow things to diffuse between. That means they have to go through cells. They have a thickened basement membrane, which is going to be more difficult to get through larger substances won't be able to diffuse as easily. And it's wrapped by paravascular feet from the astrocytes. That was from chapter 12. We talked about the different glial cells, and the astrocytes are in the central nervous system, wrapping around the capillaries. The blood-brain barrier is reduced in certain locations for functional regions. So in the choroid plexus, you need to be able to produce cerebrospinal fluid, and in the hypothalamus and pineal gland of the brain, they are endocrine or exocrine glands, so no, endocrine glands. So they have to be able to release their hormones. There you go, there is your blood-brain barrier, the astrocytes wrapping around it. You have these small little spaces that would allow for filtration. Okay, so now getting into the actual larger features of the brain, larger regions of the brain. Starting with the cerebrum, so the part that sits on top. It is an origin of all complex intellectual functions. The two large hemispheres are on the superior aspect of the brain. It's a center of intelligence, reasoning, thought, memory, and judgment, as well as voluntary motor, visual, and auditory activities. So when we talk about the brain, all of the different lobes in the cerebrum, all of the different portions of the brain have different functions, and even different of the gyri, different of the folds are going to have different functions. But it's not always as simple as saying, oh, this one folds does this. It's, like with DNA, we can say, okay, well, this sequence of DNA is this gene and it will code for this protein. It will code for this, this feature. We can't always be as specific with the brain because there are so many different parts that interact with it. So as we go through this chapter, and as we're really kind of breaking down the different portions of the brain and their functions, just try to remember that there's going to always be some other part that plays a role too. There may be one kind of large governing body in the brain that governs intelligence or taste or fine motor movements, but there's always going to be some other part that has to play a role too. So the brain never really acts in just one spot. So the cerebrum is composed of left and right hemispheres. You have a longitudinal fissure, which is a deep cleft that separates those two. At a few locations, the white matter tracts connect the two hemispheres. So remember, the white matter are the myelinated axons. They're going to be connecting the different regions of the brain. You have your corpus callosum, which is the largest tract, providing connection between the hemispheres. The regions of the cerebrum may exhibit multiple functions, some not easily assigned to one region. Again, sometimes it's hard to say, oh yeah, this is the exact spot where this happens. Connections with the body are generally crossed, meaning that the left hemisphere will control the right side, the right hemisphere controls the left side. Some higher order functions exhibit lateralization, which means they're primarily controlled by one side of the brain. Speech is frequently located in the left cerebral hemisphere. Again, this can, it's a general trend. 90 to 95% of people, I think, have speech located in the left side, but there are some people whose speech is located on the right side. It doesn't mean that it's wrong, it's just different. So here is that, again, looking at the superior aspect of the brain, the cerebrum, cerebral, again, just knowing these adjectives. So you have your left and right cerebral hemispheres. In the frontal lobe, just behind that is the parietal lobe. You have your left and right parietal hemispheres. Separating them is this central sulcus, which is the deep groove. And on either side of the central sulcus, on either side of the groove, you have the precentral gyrus and the postcentral gyrus. We'll get into those. Following the longitudinal fissure, you can go all the way back to the occipital lobe. There are 5 lobes in each hemisphere. The frontal, parietal, temporal, and occipital. Those are the ones that are kind of the outer line ones that are named after the cranial bones. The insular lobe is not visible at the surface. That's not one that you hear very much about. Its function is not really well known, and for a while it was just kind of included in the temporal lobe. So it's something slightly newer. The frontal lobe is the anterior part of the cerebrum. Its posterior border is the deep central sulcus. So as you go back from your frontal lobe, it stops at your central sulcus. The precentral gyrus controls voluntary movement, so it controls that voluntary skeletal movement. The lateral sulcus separates the inferior frontal lobe from the temporal lobe. So this is saying that the posterior border is the deep central sulcus, so go all the way back to the central sulcus, and then it will go as far down as that lateral sulcus, and then you're in your temporal lobe. The frontal lobe has varied functions: motor control, concentration, verbal communication, decision-making, planning, and personality. The parietal lobes. The anterior border is the central sulcus, so meaning the parietal lobe will come all the way up to the central sulcus. Located just behind that is the postcentral gyrus. The posterior border is the parieto-occipital sulcus. So its anterior border is its central sulcus. Its posterior border is going to be the parieto-occipital sulcus. And then lateral border is the lateral sulcus as well, dividing the temporal lobe. It serves general sensory functions, evaluating shape and texture of objects, as well as temperatures and things like that. The temporal lobe is located inferior to the lateral sulcus, and its functions include hearing and smell. Your occipital lobe is going to be for vision and visual memories, and then your insular lobe is for memory and sense of taste. So again, it's deep to the temporal lobe, and its functions used to just be attributed to the temporal lobe. Okay, so within these different lobes, within the frontal lobe rather, there are different areas that have different functional properties. So the motor areas are housed within the frontal lobes, meaning that the motor control. You have your primary motor cortex, which is located in the precentral gyrus, and it's also called your somatic motor area. It controls skeletal muscle activity on the opposite side of the body, so it projects contralaterally within the brainstem or the spinal cord. So as nerves run out through there, it's going to project across. The controlled body regions map is a motor homunculus. A homunculus is just a representation of how much cortex or amount of cortex is dedicated to a certain body part. So for instance, our hands, things that have a lot of fine movement, require a lot of fine movement, require more nerves, and so they're going to have more cortex space dedicated to them versus things that require just large, clunky kind of movements. So what we're looking at is we're saying that within the cerebrum, we have our 5 lobes. Each of the 5 lobes is going to have a slightly different function, generally. The frontal lobe is generally involved with motor functions. That's one of its things, is that it controls our motor functions. And the specific part of the frontal lobe that is generally attributed for motor functions is this primary motor cortex in the precentral gyrus. Okay, so just to go ahead and hop on this before we get too far ahead, let's make sure we know what we're talking about. Okay, spin around. So looking down at the brain, you can see here is your frontal lobe. Just behind the frontal lobe is going to be the precentral gyrus. Then you have your central sulcus, and then your postcentral gyrus, which leads to the parietal lobe. So within the frontal lobe, the precentral gyrus is part of the frontal lobe. This is where we have our primary motor cortex. So all of the nerves that are going to help regulate the motor output for skeletal voluntary movement they're going to reside in this side and on that side. I'm actually going to leave it like this because I want to have this open while we talk. Okay, so this controls skeletal muscle activity in the of the body. Yeah, we did that. So this is the homunculus that I was talking about. It's basically just saying that look at the parts that have more fine movement. They're going to require more. The larger they appear, the more space of the cortex they take up. You know, your elbow, your forearm, your knee, your ankle, they don't really require a lot of dexterous movement, so they don't take up a lot of space. But your hand, your thumbs, your face, your tongue, parts of your trunk, your toes, they have a little bit more space. Okay. So the motor speech area is the Broca area, also what it's called. It's located in the inferolateral portion of the left frontal lobe. So if you think about your left lobe, inferolateral, so out to the side portion, it controls movements for vocalization. So still a motor function, the movements for vocalization. What I'm doing right now, the regulating my breathing, being able to move my lips, my tongue, my teeth, all functioning, is the movements for vocalization. The frontal eye field is on the superior surface of middle front gyrus. So one of these gyri in here on the side of the brain, it's going to be housing the frontal eye field for eye movements. Needed for reading and binocular vision. So binocular meaning that you're looking, both eyes are observing something and giving you one image, versus monocular, which is going to give you both eyes giving you an individual image. Your premotor cortex is located anterior to, hold on, that's not right. Primary motor cortex. I think it's, yeah, says premotor cortex. Change your notes to say primary motor cortex. That's what it should say. So the premotor cortex, it's called a somatic motor association area. So you have your primary motor cortex, which is controlling a lot of the voluntary muscle movement, but then you have what's called association areas for all your functions, for both motor and sensory functions, that just help to fine-tune and regulate those larger functions. So this coordinates learned skilled activities. So things that you have done over and over again if you're a rock climber or going bicycling. A lot of us know how to ride bikes. Those kind of learned skilled activities, those get stored in our premotor cortex. So while our primary motor cortex will be the one that sends signals to move our legs and to hold our arms and to move our head the right way, it's the premotor cortex that's going to allow us to feel comfortable doing that. That's going to kind of put us into the rhythm of, no, your body knows how to do this. That muscle memory is stored in the premotor cortex. So then we get into our sensory areas. So the primary somatosensory cortex, remember when you think of somato, you're just referring to your body. So the primary somatosensory cortex is located in the postcentral gyrus of the parietal lobes. So this is our precentral gyrus. We have our central sulcus, which is the groove, and then you have your postcentral gyrus. You have one on your left, and you have one on your right. That is continuous with your parietal lobe. I don't know if we need that anymore. It receives somatic sensory information from your proprioceptors. Your proprioceptors are in your joints. They help you to understand the relative position of your joints. Your touch, pressure, pain, and temperature receptors. It also includes areas of the body sending input that can be mapped as a homunculus. So you have a sensory homunculus and a motor homunculus. Same thing, the distorted proportions reflect the amount of sensory information collected from that one region. Immediately posterior to that postcentral gyrus, so deeper into the parietal lobe, you have your somatosensory association area. It's going to integrate touch information that allows us to interpret objects by feel. That's why you can close your eyes and you can feel something and say, oh yes, that's a smooth rock, or oh no, that's a sharp knife or whatever. There is the again, this primary somatosensory cortex in the postcentral gyrus with your somatosensory homunculus. You can see your teeth, your lips, your tongue, face, hands. They have a lot more position in the cortex than something like your hip or your leg or your elbow would. Okay, so continuing with the sensory areas, you have your primary visual cortex located in your occipital lobe, and then so all of these, just so you know, are going to have an association area with them for the next couple of ones. So you have a visual cortex, your primary visual cortex in your occipital lobe, and then your visual association area that helps to integrate color, form, and memory to allow us to identify things we see. and it's surrounding the primary visual cortex. So it's still right there within the occipital lobe. It's just an association area, so it's going to be able to connect what we see to memories of what we know. The primary auditory cortex is located in your temporal lobe, and your auditory association area is also located in the temporal lobe to help interpret sounds. So again, all these association areas, they're just here to help us to interpret and to integrate current stimuli with past stimuli, with ones that we've already know, with experiences that we've already had, so that way we can be more efficient. Because just think, if every time you heard a bird chirp, if you didn't immediately know what it was, how freaked out you would be. Like, what are all of these things coming for me? Which you may still feel that way about birds. The primary olfactory cortex is also located within the temporal lobe, providing conscious awareness of smells, and the primary gustatory cortex involves taste information. So there are association areas that we're going to learn with these. There may be, we just don't know them yet, or we're not going into them. But these are the last two. So functional brain regions, there are other functional brain regions that are integrating information from multiple association areas. So you have all of these association areas. Those were the sensory association areas. You have other association areas that are going to then be sending their information that they're receiving and they're integrating to different parts of the brain, which can then collectively integrate and process that and send out information. So like I said in the beginning, when we said like, oh, this part of the brain does that, and this part of the brain does this, it gets very much more intense than that. It's not that simple. So the prefrontal cortex is the very most frontal anterior portion of your frontal lobe. It controls complex thought, judgment, personality, planning, and deciding, and it is still developing through adolescence. A lot of times in men, it is not developed fully until sometimes 25, which is why men typically exhibit a little bit more difficulty in planning and organizing and in making decisions. Well thought out decisions. I always say that as a man, and that was aware of this, that at 25, that's usually when your prefrontal cortex is fully developed. I definitely noticed a change in how I thought from 24 to 26, 27. The ways that I perceive information, it all seemed to change a little bit. So yeah, it's science, right? The Wernicke area is typically located in the left hemisphere. It's involved in language comprehension, so it's kind of in this left area. It's not in one little specific spot. It's like a circular. It's just a general portion of the brain that's uses a couple of different lobes to help interpret language. Language is very tricky. Language is a whole complex thing. We're barely going to touch on it. Okay. So that was the functional, all the gray area, basically. Now let's get into our white matter. So the white matter lies deep to the gray cerebral cortex. It's composed of myelinated groups, myelinated axons grouped into tracts. So in the peripheral nervous system, we call them axons. Here, we're calling them tracts. Same thing, okay? You have 3 different types of tracts throughout your brain, and essentially, remember, they're myelinated axons, so they're there to help transport information. The gray matter is what's going to be interpreting, processing. That's the cell bodies, the dendrites. That's what's going to be processing all the information. Whereas the white matter is going to be transporting signals back and forth to all of these areas that we were just spending time learning. It's all connected by these tracts. So the association tracts connect regions of the cerebral cortex within the same hemisphere. So in between the left hemispheres and the different parts of the left hemisphere, you have association tracts that are going to be running back and forth between the different lobes, connecting them. Then you have commissural tracts, which connect the different hemispheres, which connect the left and the right hemispheres. These include the corpus callosum, the anterior and posterior commissure. The corpus callosum is the thickest, the largest of those. You have projection tracts, which link the cerebral cortex to the inferior brain regions, the brainstem, and the spinal cord. You probably have heard of your corticospinal tract, which is carrying signals from the cerebral cortex to the spinal cord. Remember, the signals originate in the cortex and the gray matter. They're transported by the white matter. As this projection tract passes between the thalamus and the cerebral nuclei, it's called the internal capsule. That's just something you don't even really need to know. So here is just a view of that. So these are the association tracts. Staying within one hemisphere, you have these thin association tracts going between the different lobes, connecting and passing information. You have your corpus callosum, which is your association tract. Wait, did I say that right? No, commissural tract. Association tracts are the purple ones. Commissural tracts are the ones connecting between the two hemispheres, your corpus callosum. And then you have your projection tracts, which are these longer ones connecting the higher brain to the lower brain. And here, of course, I'm going to show you a representation. So, and here, if we get rid of our ventricle, and we get rid of some of these other things, that's the choroid plexus, and we'll look at this stuff in a second. Oh. Okay, so here you have your corpus callosum. So it is this large structure that's connecting the two sides of the brain. It is just all axons. Again, that's just all made up of axons, just being able to connect and send nerve signals back and forth. Okay, so that's your corpus callosum, one of those commissural tracts. All right. There we go. All right, so specialization. Cerebral lateralization. The two sides of the cerebrum exhibit differences in their higher order functions, which allows them to divide the hemispheres up into different functional regions. So they do different things. So the categorical hemisphere is the one that is used for language abilities, categorization, and analysis. So it's that we always call it the left brain. It's the very kind of analytical brain that we think of. That's on the left side. And it typically is for 90 to 95% of people. The right side, the right hemisphere, is typically called the representation hemisphere. It's what we would generally consider the more artsy, spatial relationships, imagination. And I think it's kind of misleading to think of the right side as like the artsy creative side, which that is the representational hemisphere. That's what it's for. But think about things that require, if you were very good at geometry, if you were able to really kind of orient things in space, I feel like there are a lot of people that are really talented engineers that are very right brain focused because they're able to visualize things in that way, which people that tend to be left brain focused aren't as good at. So this is not to say that you don't use your right hemisphere or you use one hemisphere less than you use the other hemisphere. It's just sometimes areas sides of the brain control things better. It's not like you have one side working harder than another. Also, while we're at it, there is that idea that you're using 10% of your brain is completely false. You use 100% of your brain all of the time. If it turns out that you're not using a portion of your brain, that's a very bad thing. That means that you have brain damage, and you should be getting treated for that. You should know about that. So this idea that, I'm left-brained, I'm going to be very, organized, I'm going to be very focused, and I'm very good at critical thinking kind of things. Okay, well, maybe I also just enjoy doing those. That's what I've really been focused on and raised on. It doesn't necessarily mean that your right brain isn't as strong, or the right side of your hemisphere, the right hemisphere of your brain isn't as strong. It just means that you haven't spent the time developing those techniques. There is a genetic component to this. There is, when we talk about, nerves being to fire and how things are set up in individual brains, there is a component that some people may be more skilled at art. They have just a better optimization in their brain chemistry and their brain patterns to be able to be artsy or to be able to be more critical and analytical thinking. But it's not that we don't have access to all parts of our brain. Okay, the two hemispheres communicate through the corpus callosum and the commissures. So lateralization develops in early childhood. You see it prior to 5 to 6 years. If a child has to have some damage to a hemisphere, if they have to have a hemispherectomy where one side is removed, the brain can regenerate itself, and the brain can even start to take over certain portions of that function. The lateralization differs between the sexes. So the women's corpus callosum tends to be a little bit thicker, so it has more axons connecting the two sides. Males tend to suffer more functional loss when one hemisphere is damaged. Because we don't have the strong of a corpus callosum, we don't have as strong of connections between the two sides, we have to have that to function better. Lateralization is also correlated with handedness. So are you right-handed or are you left-handed? And right-handers, left hemisphere is almost always categorical, speech dominant, whereas in left-handed individuals, they may have either hemisphere be categorical. So it does shift. But considering that 90% of the population is right-handed. I think 95% of the population also has the same core organization. So here is just a representation of all of that, kind of showing you the left hemisphere being categorical, right being representational, the right side controlling left-hand motor control, left side controlling right-hand motor control, the different speech areas, the motor speech area, Broca's area. Here's Wernicke's area back here, your visual fields in your occipital lobe. Yeah. Okay, so looking at the cerebral nuclei. So we did the cerebral cortex, which was the superficial gray matter that held all the functional areas. Now we're going to look at the deep gray matter. So the cerebral nuclei are the gray matter deep in the cerebrum. The only difference, and I don't want to confuse you, the difference between cortex and nuclei are just the space. It has nothing else to do with function or anything. It's just the space between them, or where they're located. Cortex are superficial, nuclei are deep. These help to regulate motor output. Disease of these nuclei are associated with involuntary movements, Parkinson's specifically. There are different parts to it. You have your caudate nucleus, which is an enlarged head and center tail paralleling the lateral ventricle. It helps to control the pattern and rhythm of walking movements. So as we're an infant, we're learning how to walk. We start establishing that pattern. And then now as adults, we can walk and we don't really have to think about that coordination of our whole body. It also contains the lentiform nucleus, which has both the putamen and the globus pallidus. The putamen helps control movements at a subconscious level, so that idea that, when you're typing, when you're texting, when you're doing different things, those learned movements that we know so well, you don't really have to think about them, as well as breathing, right? Globus pallidus influences the thalamus to adjust muscle tone. And then the claustrum processes visual information on a subconscious level, and the amygdaloid functions in mood and emotions. So just to let you look at those. Oh, I think I have to put everything back on. So we remove the dura mater, the arachnoid, cerebrum. Okay, so we have our ventricles. Then you have your caudate nucleus. You have your putamen. And then inside, so again, just so you can remember where we are. So you have your lentiform nucleus is composed of both the putamen, which is that highlighted structure right there, and then deep into that, you can see that right there is your globus pallidus. It has this has it divided into right and left segments, but these two things collectively make up your lentiform nucleus. And then you have, like I said, your caudate nucleus wrapped around it, and your amygdaloid body right there. So looking just in front of the ventricle is your amygdaloid. That's your hippocampus as well. We'll get into that next time. Okay. And I think that is going to be it for us for this lecture. Yeah, this is just showing the cerebral nuclei. So again, your cerebral cortex is the outer superficial portion. Then you have your white matter, and then you have your cerebral nuclei, which are a little bit, or rather, your cerebral nuclei here, which are a little bit deeper. These are the white matter tracts, the corpus callosum and the internal capsules.